Just dim the lights a bit so this is a bit clearer. So you get these fibrils, and they are somewhat helical in shape, as the one on the right and one on the left. The point is that new misfolded proteins get aggregated, get collected, get recruited into these fibrils, and they grow over time. Now, of course, all of our brains probably have some of these, but in Alzheimer's disease, there's an unholy number of them. Although I must say, parenthetically, that Alzheimer's is a very mysterious disease, and some people can show many of the symptoms and not have a lot of these. But anyway, as this process unfolds, you get these proteins that aggregate into regular sort of fibrils, and then when you get enough fibrils, you get the deposits of these so-called plaques against a background of normal brain tissue. So, as I said, Alzheimer's disease, not a prion disease. It's not infectious. You don't get Alzheimer's disease from caring for a loved one who has it. But it's got these commonalities, misfolded proteins aggregating into plaque-like bodies. And this is where I think the newest frontier of prion disease study is heading. Of course there's concerns about chronic wasting disease and other human prion diseases, but is there some sort of commonality between the prion diseases, which are relatively rare, and the much more common and much more serious diseases like Alzheimer's, Parkinson's, and ALS, Lou Gehrig's disease? So I would like to now introduce you to Neil Cashman. He is a Vancouverite. He's at UBC. He is Scientific Director of PrionNet, the National Center of Excellence that studies prion diseases. And Neil has really been one of the people in the forefront of looking at how the prion diseases might shed light on these other devastating neurological diseases, and he's just going to finish off the evening talking about his work. And, I've got to say, I'm interested in this stuff, I talk about it, he actually does it, and that's what's really important. Neil.
Thanks, Jay. Hopefully I can get this thing to work, to live up to your expectations.
Backwards.
Okay, so just a word about PrionNet Canada. You've heard it referred to. PrionNet is sponsoring tonight's presentation. But PrionNet is one of these networks of centers of excellence. These are federally funded, Canadian federal government funded research networks which are aimed at particular large socioeconomic problems that can really only be solved with multiple approaches, from basic science to social science. We've gone from having 2, maybe 3 prion researchers in Canada back in 2003 to almost 100, and going from a handful of students to 250. So we're helping to build the next generation to help solve these problems. I want to spend a minute talking about the prion hypothesis. Jay did an excellent job, but if you have a taste for weirdness in biology, this is a very interesting concept to wrap your head around. Instead of using animations, I'm just going to use cartoons. There's a normal cellular protein which all animals have, or at least all vertebrates have. It's most abundantly expressed in the brain, but it's virtually everywhere. It's ubiquitous. All cells and tissues in the body express this protein. The normal protein is a well-behaved cell surface membrane protein. It's rich in alpha helical content, hopefully conveyed by the bed spring. It's highly soluble in water and other solvents. It's easily digestible by the enzymes that break up proteins called proteases. It's a good guy. This amino acid chain, as Jay was showing you, all proteins are made up of a chain of amino acids folded in a particular way. This chain can be refolded or misfolded into an abnormal form. And when the prion protein is misfolded in disease, it acquires a number of distinct characteristics from the normal protein. It becomes rich in beta sheet content, hopefully conveyed by the saw teeth. It tends to stick to itself. It aggregates. It becomes resistant to the proteases, to the enzymes that break down proteins, so it tends to accumulate. But it's the acquisition of another property that really makes it epic setting in biology, and that's the ability, as Jay was saying, to recruit more copies of itself from the normal form. So when the good guy comes in contact with the bad guy, it's two bad guys that leave the scene. And you can conceive that this is an exponential process, somewhat like the mousetraps, where first there are two, then there are four, then there are 8, and on until all the prion protein in a brain or even in a body are converted to the abnormal infectious form. So I'm primarily a neurologist. I'm somebody who treats people with brain diseases. I also, of course, do work in the laboratory on prion diseases and other protein misfolding diseases. But a key question that occurs to me and somebody like me is, how do you treat these diseases? You can't use antibiotics. You can't use antivirals. The whole thing occurs on a protein-only level. There's no nucleic acid, no RNA or DNA to inhibit. So many people, including myself, have been trying to figure out if there are immunotherapies, vaccines, or passive infusion of antibodies that could work to treat these diseases in people and perhaps prevent these diseases in animals using vaccines. So this work started with the notion that if you get enough antibody directed against the normal form of the prion protein, you can cure cells of prion infection. And this has been demonstrated 20 times in vitro in cells in culture. However, when you consider a human being, do you want to infuse an antibody that's reactive with a normal cell surface protein? Well, there's another answer to that. It's called autoimmunity. You can wind up killing all the cells that have this protein at the cell surface. So this would be a very bad treatment. The treatment would be almost as bad as the disease, or maybe even worse or faster. So my colleagues and I have spent really over 10 years at this point trying to find parts of the abnormal prion protein that could be exposed, could be targeted in an immunotherapy or a vaccine. But that's a topic for another evening. I want to say that prion diseases and diseases like them can be regarded as a huge challenge, but also as an opportunity. Jay showed you that mice inoculated with Alzheimer's disease brain can come down with Alzheimer's disease. But what can we tackle? What can we approach? How could we possibly treat Alzheimer's disease? Well, it turns out that antibodies directed against the implicated protein, which is called A-beta, can block Alzheimer's disease in mice. Vaccines directed against A-beta are actually reaching human treatment trials. So this is an idea whose time has come. Parkinson's disease, if we live to 85, say, about half of us develop Alzheimer's disease, about 10% of us develop Parkinson's disease, another very common neurodegenerative syndrome. The third one, as Jay was saying, amyotrophic lateral sclerosis, or Lou Gehrig's disease. About 1 out of 1,000 of us died of that disease. Over the past few years, it's been recognized that transplanted neurons in the brains of Parkinson's patients also develop Parkinson's disease. And this is shown by the acquisition of a pathological marker called a Lewy body. This is made up of a protein, alpha-synuclein. So the healthy transplant in the Parkinson's disease brain acquires Parkinson's disease. How does that happen? It's likely through the spread, the cell-to-cell spread of alpha-synuclein misfolding, converting this normal transplanted cell into a cell that is now sick with Parkinson's disease. Again, this provides us with an opportunity, maybe even an Achilles heel of Parkinson's disease. If we can immunize people or infuse antibodies against misfolded alpha-synuclein, then we could potentially block or at least slow this disease. The disease I've spent the most time working with in the clinic and in the laboratory is ALS, the Lou Gehrig's disease. We have identified a prion-like activity of another protein called superoxide dismutase 1. We can show that SOD1 misfolding can be transferred from cell to cell, and that we can block the progression of ALS in a mouse model using antibodies against this misfolded protein. So prion hypothesis, expanding. I've got a question mark, which is we still don't know for sure. It's early days with this group of diseases. This whole concept is about 10 years old. I've also got an exclamation point to convey that we're in trouble here. If Alzheimer's disease is contagious, if Alzheimer's disease is infectious, like the prion diseases, then we've got a whole new raft of problems to face. The good news is it probably is not contagious from person to person unless there are very specialized circumstances. For example, neurosurgical instruments going from somebody with early Alzheimer's to somebody who doesn't have early Alzheimer's. There will be new health concerns for infectivity. We need to fish out if there is a problem, if there is risk in neurosurgery or organ transplantation or maybe even blood transfusion. But we are also facing an opportunity, which is the opportunity to stop the cell-to-cell spread of protein misfolding, which may, in fact, arrest clinical progression of disease, to stop Alzheimer's, to stop Parkinson's, to stop ALS from progressing. So that's all I wanted to say, and I'd like to say Jay and I will be very happy to answer your questions. But if you have a bigger question, if you have some reason to contact us at UBC to interact with us at PrionNet Canada, we would be absolutely delighted. So I thank you for your attention, and Jay, why don't you come back up here?